Hello friends, Maniepa here, and biology paper 2 theory is what's up this morning or whatever time you're watching this video. NS at heart is what this is all about, because for the love of science we do these things. We straight away get to question 1. I'll just do section A for this paper, then I'll do section B later. So figure 1.1 and 1.2 show freshly cut potato chips suspended in solution E and F at the beginning of an experiment and after 2 hours respectively. That's 1.1, 1.2. Fresh, they are equal amounts of solution E, solution F. Yes, potato chips at the beginning. After 2 hours, solution E, solution F. Yes, they appear to be of equal amount, but the size of the potato chips, this one is bigger, this one is smaller compared to their initial sizes. So A1, explain the difference in the appearance of potato chips in solution E and F after 2 hours. So I've explained for solution E, the chip in solution E, and then the chip in solution F. So chips or chip, okay, I just wrote chips there. So chips in solution E lost water to the solution by osmosis because the solution was hypertonic. This is solution E, and the chips or the chip in there, it lost water, it shrunk, okay, it lost water by osmosis. And this can only happen if the solution in which you have put the chip is hypertonic. Therefore, hypertonic means a solution that is more concentrated compared to the other one. That other one is the cytoplasm. So the cytoplasm will lose water to the more concentrated solution, which is referred to as being a hypertonic solution. The second one, chips in solution F gained water by osmosis from the solution, because the solution F is hypotonic. Hypotonic is another way of saying dilute. Therefore, you cannot just say hypotonic if you're not comparing 2 or more solutions. So when you check out the potato chip in solution F, its initial size and its final size, here it has really gained size, and this is because of gain of water by osmosis, because the potatoes are not boiled. If they are boiled, then nothing will happen because protoplasm would be dead, the cells would be dead. So this only happens if the cells are alive, therefore the cell membrane is intact and functional. So the loss is by osmosis, the gain is by osmosis, and then the reason is because in this scenario, the cytoplasm is more concentrated and the solution F is dilute or less concentrated. So water, when it comes to concentration and osmosis, water always moves from the region where it is in high concentration to a region where it's in lower concentration across the semipermeable membrane when it's in osmosis. That is why in some cases osmosis is referred to as diffusion of water, because it's all about from high to low concentration. And then it's called osmosis because there's a cell membrane involved. The next one there, number 2, based on the appearance of the potato chips in solution E after 2 hours, what term could be used to describe solution E? Solution E is hypertonic. Okay, when you look at this one and then look at this, it's hypertonic because the potato chip loses water to the solution. This happens if you, for example, wash for some time, wash clothes for some time, your hands wrinkle. Okay, that can also happen if you were to swim in the ocean for quite some time, because this ocean water is salty, so you will definitely lose water to the salt water, meaning you will lose water to the more concentrated solution. Water moves to the more concentrated solution. Number B, B1, what would be the state of the cells in the potato chips in solution F after 2 hours? Therefore, that's our F, the one that gained water. The cells would be turgid, T-U-R-G-I-D, turgid. By turgid would mean they'll be fully inflamed with water, they'll be swollen with water, and that is referred to as being turgid. 2, state the importance of the state of cells in B1 above to plants. It aids or helps plant young stems to stand straight or to stand erect. Okay, meristems to stand erect. Even leaves to also be firm and erect. Therefore, this will help the plant to properly be oriented towards light and photosynthesize properly. If the plant loses excessive water, it won't really be turgid. It will actually wilt, and that is not so healthy. If allowed to prolong, it can bring about cell death in plants. See, explain the meaning of the term isotonic solution. Okay. Isotonic solutions, though they have said solution, but allow me to just generalize it. Isotonic solutions are of the same, concentration. Okay, that's that. Are of the same concentration. And isotonic, there is isotonic, hypertonic, hypotonic. These terms are usually used to relate two or more solutions. So if you say isotonic solution, it means a solution of equal concentration in relation to another one. Okay, so I'll get to question 2. Question 2, 2A. Okay, so question 2, the question reads, Table 2 shows various food tests carried out by a learner. Starch, and then there are gaps there. Complete the table by filling the spaces. Starch, you use iodine solution. The color changes to blue-black, then starch is present. Emulsion, white precipitate, or a cloudy appearance forms. If it forms, then lipids. In short, the emulsion test is the one used to test for lipids. And if it happens that the emulsion forms, or a white PPT, small droplets, small lipid droplets are the ones that appear to be precipitates here. And you usually have to add ethanol for this to happen. So the conclusion is that lipids are present once the white PPT is formed, white precipitate. Protein test use burette reagent. A purple color forms from blue. Therefore, burette is blue, and then it will turn purple if at all you add it to a solution that has proteins in it. It means proteins are present. Number 2, which nutrient in the table would cause kwashiorkor if lacking in the human body proteins? Kwashiorkor is a condition in which is most prominent in children because children are just fed. And if you're not careful of the type of food you're feeding your child, you may expose them to some of these dietary ailments. Adults eat so many things as they move about, so they rarely experience these. But kwashiorkor is a disease in which hair growth is not so good. The skin peels off. Okay, the limbs appear swollen, and wounds don't easily heal such things. So at the end of it all, the swelling in the limbs is simply caused by the inability of blood plasma, which is now in the state of tissue fluid, the inability of tissue fluid to flow back in the blood vessels and then travel back to the heart. So it collects in the limbs. It collects and fails to climb back to the heart. It's the flow back of tissue fluid, which is mainly water in form, is simply promoted by the presence of proteins in blood. So if you have less blood proteins due to lack of proteins in your diet, then osmosis won't easily occur. The water won't easily be regained. Of course, there are other factors that help tissue fluid to flow back into blood vessels at the end of the capillary networks, like muscle contractions and the like. So that's that, protein. Number 3, name the nutrient are not in the table which helps to prevent constipation. Roughage, sometimes referred to as dietary fiber or just fiber. B, stage 2 deficiency diseases in plants. We have leaf chlorosis and stunted growth. You can think of any other. We've scored 8 marks. Figure 3 shows a longitudinal section through a root tip. Okay, that's our root tip vascular bundles G, H, I, J. Okay, identify the parts labeled G and J. That's G here and J. This is our root hair. G is the root hair or root hair cell. Then J is root cap. Okay, the root cap, a tissue that simply prevents damage to the root as it penetrates through soil. B, which labeled part in figure 3 is responsible for generating new cells? It's I. Okay, I is responsible for generating new cells like rapidly, which eventually or later on differentiate. C, region H is responsible for cell differentiation. Apart from vascular bundles, stage 2 other cell types differentiated by region H. That is our region of cell differentiation. There is usually a region of rapid cell division by mitosis, of course. Then there is region of cell differentiation. Then there's always a region of cell elongation. Okay, they differentiate, then they grow in size as they different. After they pick pathways, they differentiate, then grow. Cell elongation, but cell elongation here hasn't been shown. So root hairs, they haven't limited this. Otherwise, they've just limited us. Let's not talk about the vascular bundles. So which other types of cells result? You've got root hair cells, okay, and then apart from that, parenchyma cells. Okay, apart from that, you can even talk about epidermal cells and many other tissue that you may figure out, but these are the ones I could think about: root hair cells and parenchyma. Stage 2 types of cells found in the vascular bundle: phloem and xylem. Phloem cells and xylem cells. Of course, at maturity, these die. At maturity, these lose a lot of their organelles, such that they are aided to function by companion cells. Number 3, which named type of cells in C2 allows movement of substances up and down depending on the needs of the plant? The phloem cells. The phloem vessels move, or maybe the contents of the phloem vessels or phloem sap moves both upwards and downwards, and that is actually referred to as what? Translocation. Number D, I didn't solve B, D. Okay, I'll leave this as your assignment. Open up 1 or 2 books. I think I wanted to check this one later on, but unfortunately I didn't solve it. Okay, I'll leave this as your assignment. Suggest one meristematic structure in the diagram responsible for bringing about secondary growth in the dicotyledonous plants. Okay. Secondary growth. What does secondary growth involve? Secondary growth involves generation or occurrence of these. Okay, primary growth involves just elongation, the shoot tip, the root tip, but secondary growth involves increase in girth or thickness of the plant, formation of new vascular bundles and the like. So if anything, the answer here should be the vascular cambium. The vascular cambium, I would say, is tissue that divides and from which the vascular tissue results. Okay, vascular tissue results. There are a number of, I think, 3 or so types of cambiums, but you can check them out later. But vascular cambium contributes to secondary growth in dicotyledonous plants or plants with two cotyledons. Okay, like avocado, mangoes, beans. You can split the seeds into 2 halves or 2 cotyledons. Question 4. In front of us is Z nephron. Check it out. KLM. Therefore, efferent artery, glomerulus, Bowman's capsule. Not Bowman's Lusa something. The first convoluted tubule or the proximal convoluted tubule, the loop of Henle. Okay. Then the second convoluted tubule, aka the diastole. Then the collecting duct, which takes urine to the pelvis. Then from the pelvis of the kidney, it flows via the ureter to the urinary bladder, then out through the urethra to the environment. So number A. Identify the parts labeled K and L. K is the afferent artery. Okay, L is the glomerulus. Okay, glomerulus, a bunch of blood capillaries that are coiled inside there. So this one is the afferent with an A. Then this one is the efferent. Usually this one is larger in diameter than this one, and because of having a large diameter, it creates high pressure here where the blood vessel becomes smaller, and it brings about what we call ultrafiltration or pressure filtration. A number of components are simply filtered from blood into the glomerulus or the Malpighian capsule. The whole of this together is called the Malpighian capsule, and as a result, what is filtered out of blood is called the glomerulus filtrate, which flows in this tubule. Some of the filtered substances are still important to the body, so they are reabsorbed into the bloodstream in these capillary networks. They are reabsorbed and flow back into the blood system because they are essential. Okay, but some are allowed to flow out in large quantities and they actually make up urine. So B1, name the substances present in fluid K but absent in fluid M. Let's check out fluid K, that is actually blood. Then fluid M, this is glomerulus filtrate in the proximal convoluted tubule. To be convoluted just means to be coiled up, the way they are coiled up. So here I had a number of thoughts, because if you look harder, when it comes to reabsorption of materials in the glomerulus filtrate, a little bit of urea is reabsorbed. Okay, so you cannot easily say there's no urea in the blood that flows back. There is a little bit of it. Okay, and then when it comes to things like glucose, a very large quantity of glucose is reabsorbed. Okay, out of the glucose, for example, which is filtered out in the amino acids, a very large quantity of them is reabsorbed back into blood. But a minimal amount of glucose remains as part of urine. So there's no such thing as 100% absent, like 100% no glucose, 100% no amino acids, such kind of thing. There's always these small variations that are within normal limits. But beyond certain normal limits, it's not normal to have a certain quantity of glucose or amino acids or even blood cells in your urine. So I had a little bit of thoughts here when they say, name two substances present in K and absent in fluid M. I was thinking of what is it that could typically be present in K, and be absent completely in the urine or in M. So I went for blood cells. Yeah, blood cells, you will not find them in M, that's definite. This one is just an alternative answer. Blood proteins also, fibrinogens, okay, and other prothrombin, those blood proteins, you don't find them in urine under normal circumstances. That was my thinking. So you can just pick one of these. My second one was glucose. It's not normal to find glucose in urine, but if you really test, you find that it's normal to find very minute amounts of glucose. But I guess those details are simply ignored for now. So glucose shouldn't be present, amino acids should be present, blood proteins shouldn't be present. Therefore, they are present in K, but they're not supposed to be present in M. That was my take there. So I've put 3 answers. Only put 2 if you're told to put 2 here. So 1, 2, leave out 1. Number 2 here, give 2 reasons for your answers in B1 above. For number 1, the blood cells or blood proteins, let me just put brackets here, they are too large to pass through the glomerulus. Okay, they are too large to be filtered through the glomerulus. Number 2, it is reabsorbed back into blood. That is why it is absent in urine. It is reabsorbed back into blood. But of course, 96% of it is what is reabsorbed. That is being critical, but the thing is, it is reabsorbed, that is why it is absent. Then these, or the blood proteins, are absent from M because they're too large to be filtered through the glomerulus. Then number C, state what common disorder associated with the kidney. I was almost rushing for diabetes. Diabetes affects kidney functioning, but again I realized diabetes actually has more to do with insulin and the like, so I just went for kidney failure. Kidney failure is an umbrella, or maybe encompasses all conditions that kidneys get to go through such that they fail to perform this filtration function of theirs to simply produce urine. So kidney failure, explain how the disorder can be treated. I was thinking of treatment. Is it all about treatment or all about can be solved or resolved? If it's treatment, I was thinking of diabetes. There are 2 types of diabetes. One brought about by maybe a diet which is not so good, okay, and depending on the age and body weight of someone. And there's also diabetes where the pancreas completely malfunctions, which can last for a lifetime. So when I thought of treatment, anyway, I went for kidney failure. which is a little bit broad, okay. And then the only treatment to this is kidney transplant through surgery from a suitable donor. The donor has to be suitable, or dialysis, but this one I've put it in brackets, so ignore this and just pick this other one. But the other one is dialysis. If you pick the dialysis pathway, you should be able to explain a little bit more of how dialysis is done to treat the disorder. So kidney failure has got many symptoms because it depends on what has caused the failure. Is it an accident? Is it some kind of, you are trying to commit suicide, you took some poisons, and now that you survived, the kidneys and the liver have, suffered the effects of the drugs, or you're just a drug addict or something. And then that would depend. And usually, if you're having kidney failure, you can tell from the urine that you're passing. Is there blood in urine? Is urine having foam? Like as you're urinating, you are seeing bubbles. Is there too much sugar in the urine? Okay. Are you frequenting like the bathroom, too much, like beyond normal ways of frequenting the bathroom as before? So there are many ways in which kidney failure can happen. But the one permanent solution could be, or treatment, is kidney transplant through surgery from a suitable donor. And I think they will give us marks, like 3 marks here. So we would have scored our 9 marks from that question. Question 5 shows 4 animal cells at different stages of mitotic division. 1, 2, 3, 4. Okay, cell 1, cell 2, cell 3, cell 4. These cells are at different levels. This is not an order of, say, from this stage to stage 2, stage 3, stage 4, because at the beginning I kind of became confused, because I was almost seeing them as if it's an order, like from this stage it goes to this, then goes to this. So like they seem to be confused, like arranged in an order which doesn't really make sense. So you should have studied mitosis, cell division or cell reproduction, and you should have understood the stages such that you can remember when you see these stages of these 4 different cells. So name the structures labeled N and O. Oh, these are chromosomes. These are actually chromosomes, okay? Oh, that's your nuclear membrane or nuclear envelope, okay? Number B, at which stages of mitotic division are cells 1 and 3? 1 and 3. What? Did I just solve for 1 or for 3? Just a moment, let me just confirm. Cool. I think I'm okay. I'm okay. I was almost thinking I went off-road. So C, a husband and wife were heterozygous for blood group A and B respectively. You have to understand what heterozygous means. They're not homozygous, meaning they're partly A or B and the other side is O, which, with the help of the genetic diagram, show the possible blood groups of their children. You can use this, or you can use the Punnett Square. It's the same thing. So these are the parents. They have said wife, so this is husband and wife, A and B. So this is husband, that's wife. This is husband, that's wife. Heterozygous, meaning this is the scenario. If the husband was homozygous A, he would have been like that. Okay, homozygous A. Same, homozygous B is actually BB. So this would be our genetic diagrams, and these would be the genotypes. This is going to be referred to as blood group AB. This would be referred to as A, just like the father. This would be referred to as B, just like the mother. So B is dominant over O. So it is usually just referred to as B. And this is actually O. Allow me to go back a little bit on these anaphase and metaphase phases of mitosis. There's this acronym that sometimes maybe may help: prophase, metaphase, prometaphase, anaphase, and telophase. This looks like promient. Okay, I don't know, you should remember this. So prophase is where the chromosomes condense after they have already divided. They condense and become more visible. They become more visible. And then there's what you call prometaphase, which is between prophase and metaphase. Prometaphase, the chromosomes begin to migrate, going to the equator of the cells. Okay, the equator of the cell, or the central part of the cell, to line up at the the metaphase, to form the metaphase plate. Okay, they begin to migrate. At metaphase, they are actually at the at the equator. That is why I had to give this as metaphase. But again, in this picture, I'm not so sure if they are aligned at the center or this is prometaphase, because prometaphase, they should be migrating towards the center of the cell. At metaphase, they have arrived at the metaphase plate, or they are all aligned at the center. Now this diagram is small and the chromosomes are big, so I began checking out these to my, this is centromere, 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 centromere. They seem to be at the center, okay, awaiting separation like in this other cell. Okay, so metaphase is when they have arrived and they have aligned themselves at the equator. And then after this, you've got anaphase. Anaphase, they are dividing, they are being pulled towards the pores of the cell, on the two pores of the cell. This is the one pore and that's the other pore. Then the telophase is where the nuclear membrane forms around the two groups, like the one that this cell is in. After telophase, what follows is called cytokinesis, okay, the pinching of the cytoplasm, which is actually this cytokinesis, where the cell pinches off or cleaves, okay, the cleavage of the cell to simply become or to form two independent daughter cells whose genetic makeup is same as the parent cell. So go through this and then compare with my solution here, and then that's that. So from this end here, the next part is going to be section B of this paper, and we'll check it out next time in another video. So from me to you, I say bye bye for now, and proofread the answers and check out and leave your comments if any, and bye bye for now.